Bismillahirrahmanirrahim. Welcome back to my channel. In this video, I'll discuss mechanisms of transport of materials across cell membrane. Transport of materials across the plasma membrane is essential to the life of a cell. Certain substances must move into the cell to support metabolic reactions. Other substances that have been produced by the cell for export or as cellular waste products must move out of the cell. The cell membrane is a barrier to free movement of materials in and out of the cell. Membrane's lipid bilayer has tightly packed phospholipids, making a hydrophobic interior. This structure causes the membrane to be selectively permeable. The term permeable means that a structure permits the passage of substances through it. The lipid bilayer portion of plasma membrane is highly permeable to non-polar molecules such as oxygen, carbon dioxide, and steroids. The term impermeable means that a structure does not permit the passage of substances through it. Membrane is impermeable to ions and large uncharged polar molecules such as glucose. The permeability of the plasma membrane to different substances varies. Plasma membranes permit some substances to pass more readily than others. This property of membranes is termed selective permeability. Membrane is moderately permeable to small, uncharged polar molecules such as water and urea. Transmembrane proteins that act as channels and carriers increase the plasma membrane's permeability to a variety of ions and uncharged polar molecules that cannot cross the lipid bilayer unassisted. Channels and carriers are very selective. Each one helps a specific molecule or ion to cross the membrane. All substances that move through the membrane do so by one of the two general methods, which are categorized based on whether or not energy is required. Passive transport is the movement of substances across the membrane from area of higher concentration to lower concentration without the expenditure of cellular energy. In contrast, active transport is the movement of substances across the membrane from area of lower concentration to higher concentration using energy from adenosine triphosphate. Passive transport involves processes, namely diffusion, facilitated diffusion, osmosis, and filtration. These types will be discussed in detail in upcoming part of this lecture. Before we discuss the differences between the diffusion and the facilitated diffusion, let's take a look at some general properties of diffusion. In diffusion, the random mixing of materials in a solution occurs because of the particle's kinetic energy. The solutes which are the dissolved substances, and the solvent, which is the liquid that does the dissolving, both can undergo diffusion. The greater the difference in concentration between the two sides of the membrane, the higher the rate of diffusion. For example, the air sacs of the lungs have a large surface area available for diffusion of oxygen from the air into the blood. In some lung diseases, such as emphysema, reduction in the surface area occurs. This slows down the rate of oxygen diffusion and makes breathing more difficult. Larger the membrane surface area available for diffusion, the faster the diffusion rate. Diffusion across a cell membrane takes only a fraction of a second because the membrane is so thin. In pneumonia, fluid collects in the lungs. The additional fluid increases the diffusion distance because oxygen must move through both the build-up fluid and the membrane to reach the bloodstream. The higher the temperature, the faster the rate of diffusion. All of the body's diffusion processes occur more rapidly in a person with a fever. The larger the mass of the diffusing particle, the slower its diffusing rate. Smaller molecules diffuse more rapidly than the larger ones. Simple diffusion is a passive process in which substances move freely through the lipid bilayer of the plasma membrane of cells without the help of membrane transport proteins. Non-polar hydrophobic molecules move across the lipid bilayer through the process of simple diffusion. Such molecules include oxygen, carbon dioxide, and nitrogen gases. Fatty acids, steroids, and fat-soluble vitamins also cross the membrane by simple diffusion. Substances that have large size, bear charge, and polarity cannot cross the lipid bilayer. Facilitated diffusion is a process by which molecules are transported across the plasma membrane with the help of membrane proteins. Facilitated diffusion is having two varieties based on the type of transport proteins in use. The two types are channel-mediated and carrier-mediated facilitated diffusion. The channel is an integral transmembrane protein that allows passage of small inorganic ions that are too hydrophilic to penetrate the non-polar interior of the lipid bilayer. Channel proteins do not bind with the ion which is being transported. Each ion can diffuse across the membrane only at certain sites. In typical plasma membranes, the most numerous ion channels are selective for potassium ions or chloride ions. Diffusion of ions through channels is generally slower than free diffusion through the lipid bilayer because channels occupy a smaller fraction of the membrane's total surface area than lipids. Still, facilitated diffusion through channels is a very fast process. One type of channel proteins are open at all the times. They are called leak channels, constantly allowing a particular substance into or out of the cell depending upon the concentration gradient. Second type, called gated channels, are closed most of the times and can only be opened by a particular biological signal. Gated channels are further classified on the basis of the type of biological signal that opens them. Signal for voltage gated channels is a change in transmembrane potential. Ligand gated channels open when a specific chemical binds with the receptors on the channel. And signal for mechanogated channel is a mechanical change in the environment. The carrier is an integral transmembrane protein that allows passage of large polar molecules. Carrier proteins aid in facilitated diffusion by binding a particular substance, then altering their shape to bring that substance into or out of the cell. A common example of facilitated diffusion is the movement of glucose into the cell, where it is used to make ATP. Although glucose can be more concentrated outside of a cell, it cannot cross the lipid bilayer via simple diffusion because it is both large and polar. To resolve this, specialized carrier protein called the glucose transporter will transfer glucose molecules into the cell to facilitate its inward diffusion. The number of carriers available in a plasma membranes are limited. Once all of the carriers are occupied, there is no further increase in the rate of facilitated diffusion even after an increase in the concentration gradient. This upper limit is called the transport maximum. Thus, the process of carrier-mediated facilitated diffusion is said to exhibit the phenomena of saturation. An example of this process occurs in the kidney. Glucose, water, salts, ions, and amino acids needed by the body are filtered in the glomerulus. This filtrate, which includes glucose, is then reabsorbed in the proximal convoluted tubule of nephron. Because there are only a finite number of carrier proteins of glucose, when more glucose is present than the proteins can handle, the excess is not transported. Rather, it is excreted from the body in the urine. Hence, in a person with diabetes mellitus, this is described as spilling glucose into the urine. It is pertinent to remind that the type of carrier protein in proximal tubule of kidney mentioned above is different from the group of carrier proteins called glucose transport proteins or glutes, which are involved in transporting glucose and through plasma membrane within the body cells. Osmosis is the diffusion of water through a semi-permeable membrane. Osmosis occurs only when a membrane is permeable to water but is not permeable to certain solutes. In this scenario, water will equalize its own concentration by diffusing to the side of lower water concentration and thus the side of higher solute concentration. During osmosis, water molecules move across the membrane in two ways, either by moving between neighboring phospholipid molecules via simple diffusion or by moving through integral membrane proteins that function as water channels and are called aquaporins. Cells have mechanisms to regulate the movement of ions or solvents, but the movement of water molecules is not itself regulated by some cells. So, it is important that these cells are exposed to an environment in which the concentration of solutes outside the cells is equal to the concentration of solutes inside the cells. Two solutions that have the same concentration of solutes are said to be isotonic. When cells and their extracellular environment are isotonic, the concentration of water molecules is the same outside and inside the cells, and the cells maintain their normal shape. Osmosis occurs when there is an imbalance of solutes outside of a cell versus inside the cell. A solution that has a higher concentration of solutes than other solution is said to be hypertonic. And water molecules tend to diffuse into a hypertonic solution. Cells in a hypertonic solution will shrivel as water leaves the cells via osmosis. In contrast, a solution that has a lower concentration of solutes than other solution is said to be hypotonic, and water molecules tend to diffuse out of a hypotonic solution. Cells in a hypotonic solution will take on too much water and swell with the risk of eventually bursting. A critical aspect of homeostasis in living things is to create an internal environment in which all of the body's cells are in an isotonic solution. Various organ systems, particularly the kidneys, work to maintain this homeostasis. Another mechanism to passively transport materials between compartments is filtration. Filtration uses pressure gradient of the fluid, also known as hydrostatic pressure, to push the fluid and the solutes within it from a higher pressure area to a lower pressure area. Filtration is an extremely important process in the body. For example, the circulatory system uses filtration to move plasma and substances across the endothelial lining of capillaries and into surrounding tissue, supplying cells with the nutrients. Filtration pressure in the kidneys provides the mechanism to remove waste from the bloodstream. Thanks for watching and listening. Please do share this video with your friends and colleagues.